But next up, as I move through, we will have the cancer-epitope database and analysis resource, also known as CEDAR. Very exciting new project that's been kicked off by the team. And I would now like to introduce Dr. Zeynep Koşaloğlu-Yalçın to present CEDAR. Thank you very much, Zeynep.
Yeah, thanks, Nina, for the introduction. So as Nina already mentioned, I will be talking about the cancer-epitope database and analysis resource. So this is a very new exciting project that we started earlier this year. But I want to start with a brief introduction into cancer antigens and epitopes. So there are a lot of different cancer antigens available, and they are usually classified according to the expression patterns of the parent gene. So starting here on the right, we have overexpressed antigens. So some genes, for example, genes that play a role in oncogenesis, are often overexpressed in some tumors. So the resulting antigens will then be highly overexpressed in tumors, but they can also be expressed on some healthy cells. So these antigens are not tumor specific. So then we have differentiation antigens. They are derived from proteins with tissue-specific expression. So they are only expressed in a specific tumor type. So in this case, for example, melanoma, and in the corresponding healthy tissue, in this example, skin cells. But these antigens will not be presented on any other healthy cell. So tumor-specific antigens are encoded by cancer germline genes. So these genes are only expressed in tumor cells as a result of demethylation, and also in germline cells, as the name says. But the resulting antigens will only be presented on the tumors because the germline cells do not express HLA. And these antigens will also not be present on any other healthy cell. And then we have antigens that arise as a result of mutations, so-called neoantigens or neoepitopes. So a hallmark of cancer is that they accumulate mutations in their genome. So that's shown here in green. And when the mutation is translated, it can generate a mutated peptide. And then if the mutated peptide presented, it will be seen as a new antigen with a mutation within the sequence. And because the mutation is only present in the cancer DNA and not in the normal DNA, the normal cells won't be presenting these new antigens, but they can present the associated wild-type peptide, for example. So these new antigens are very interesting because they are highly tumor-specific. So all of these different cancer antigens are being widely studied, some of them for decades, actually. So by now, it is well appreciated that cancer antigens and cancer epitopes in general play a very important role for cancer immunology. Cancer epitopes have been proven to play an important role to understand why some patients, for example, respond to therapies better than others. Cancer epitopes can also be used as therapeutic targets, for example, as vaccines or for other cell-based therapeutic approaches. So there's a clear need in the community to catalog all of these cancer epitopes so that we can study them better and understand them better. And this is also evident by the fact that in recent years, a lot of different databases were published. They all attempted to do the same to catalog cancer epitopes. But when we had a closer look at those epitopes, at those databases, we found that the data is not captured in as granular of a fashion as we would like it, meaning linked to all the biological, immunological, and clinical contexts. Also, importantly, the existing resources only provide very limited computational tools to perform analysis on the captured data and to perform predictions, for example. So to address all these gaps, we developed the cancer epitope database and analysis resource, which we abbreviated CEDAR. So as we've already heard earlier during the workshop, the IEDB is a main resource for everything epitope related. But I want to mention here again, importantly, that the IEDB hosts epitope data only for allergy, for infectious diseases, for autoimmunity and transplantation, but cancer is not included in the funding of the IEDB. So this is why we applied for a separate grant at the National Cancer Institute, and we received funding earlier this year to extend what we have been doing for the IEDB and develop a similar resource specifically for cancer epitopes. So just like the IEDB, CEDAR also has two components: the database, where we curate the epitopes from literature, and an analysis resource, where we provide bioinformatics tools to analyze the captured data and also to perform predictions. So we have four main goals for the CEDAR project. First, we want to establish the database and the underlying ontology, and to develop a cancer-specific query and reporting functionalities. Second, we need to curate cancer epitope data from the literature and to populate the established database. The third aim is related to the analysis resource component. Just as we are doing in the IEDB, we will provide a set of tools that we have validated for predicting and analyzing cancer epitopes. And we also want to engage the cancer research community to get feedback and also to develop features that are needed in the research. So before we started curating cancer epitope data, we first needed to define which elements we need to accurately capture such data. So we have defined 7 main elements that are necessary to accurately describe the epitope together with its biological and clinical context. So namely, we have the epitope sequence, the source antigen, information about the host and the tumor sample. We have details about the experimental assay that was used to describe the epitope and the immune cells that were involved, and also details about the study that published the epitope. So for each epitope we curate and capture from the literature, we try to capture all these details whenever they are available. So of all the papers we have identified as containing epitope data, around 11% are cancer related. So right now we have just over 3,500 papers identified as containing cancer epitope data. And we have an internal queue for curation. So we started with curating new epitopes, given that they are highly interesting right now. And we also started curating prostate antigens recently. So the neoepitope curation, we actually already started last year as part of a pilot project for the NCI. So right now we have curated almost 250 papers, and we are curating also new papers as they are being published. So CEDAR is not online yet, it's still in the development stages, but we have generated this mock-up of how we envision the homepage and query interface to look. So it looks really similar to the IEDB interface. And we have also some elements that we also know from the IEDB, for example, here the epitope, the MHC restriction, or the assay. But we also needed to add some more fields to enable really cancer-specific queries. So an example here, for example, we have neoantigens, and the user will be able to put in a specific mutation and query for all the neoantigens associated with that mutation. Or, for example, you might want to search for epitopes associated with a specific cancer type. For example, you can search for breast cancer and get all the epitopes that were published in context with breast cancer. Or here, one interesting element is the treatment. So you might want to search for epitopes from patients that received a specific kind of therapy. So for the analysis resource part of CEDAR, we want to provide prediction and analysis tools specifically for cancer epitopes. So there are already a lot of tools and pipelines published, but those tools are often quite difficult to use because they require some programming experience to even install and also to use. So what we want to do is we want to curate a list of such tools based on their functionalities they offer and also how often they are used and requested in the community. And once we have identified the tool to be valuable and that we are able even to implement it, we want to provide a web accessible version of the tool in the CEDAR analysis resource, so where they can be then easily used with no programming experience. So there will be tools that we already have also in the IEDB, such as an MHC pen to perform binding predictions. But we have also identified some cancer-specific tools, for example, tools that calculate the foreignness or the self-similarity of a given peptide, or tools to estimate the tumor fitness from a given patient sample. So we will also provide tools that are specifically needed by cancer researchers. So one common question, for example, is what neoepitopes are generated by a given mutation? So to answer that, the user will be able to input a mutation in different formats, which then our tool will take in and then translate to mutated peptides. And those peptides then can be further used, for example, to perform binding predictions. And it will also be possible to perform side-by-side predictions. So for the mutant and the wild-type peptide together, so that the user can then compare the results. And we also want to use the cancer-epitope data that we are curating in CEDAR to develop novel tools. So currently, epitope predictions are mainly based on HLA binding predictions. But recent studies by us, for example, and also other groups have shown that epitope predictions can be improved by combining binding predictions and the RNA expression levels of the associated antigens. So we want to use the data curated in CEDAR to explore which additional features might be beneficial to further improve predictions. And ultimately, we want to develop a novel machine learning method that takes into account all the different features that we have identified and hopefully provide more accurate predictions. And last but not least, we also want to use the curated epitope data to benchmark existing tools and also the novel tools that we are developing. So we will run benchmarks manually in the beginning, but ultimately we want to set up an automated benchmark server that automatically performs benchmarks in certain intervals for all the tools implemented in CEDAR, similar to what we are also doing in the IEDB right now. So to perform a useful benchmark, it is important to clearly define the rationale of what is being predicted. And then the associated benchmark data sets also have to be assembled accordingly and carefully. So we have here two examples, what could be the rationale of such a benchmark. For example, what peptide in the tumor sample are processed and presented on MHC, or what neoepitopes are recognized by T cells from a cancer patient. So these are quite different questions, and the benchmark data sets also have to be then curated accordingly. But importantly, we also, once we have assembled such data sets, we will provide them as benchmark data sets in a simple format to the community so that other bioinformaticians and groups can easily use them to train new tools and also to test their tools. So to summarize, we started developing CEDAR, which will be an extension of the IEDB and will contain cancer-related epitope data and tools. We will adapt existing tools from the IEDB so that cancer researchers can easily use them for their projects, and we will also develop new tools. CEDAR is not online yet, but we are planning the first release next year, so keep an eye out for that for sure. And yeah, with that, I want to thank you for your attention, and feel free to drop any questions in the Q&A so that we can talk about it later. And yeah, so I'll hand it over to Björn, I think, for the next presentation.
Yes, thank you so much, Zeynep. I really hope everyone enjoyed the behind the scenes insight into CEDAR. This is very exciting and something that has been a long time coming as well. So we're really excited to get that online for you.